our program for today is very practical, hands-on, but very, very fun. As you're seeing the videos, we have some alien-shaped stuff in the left. We'll figure out what it is. In the middle, we have another weird thing. We call that macrophages chasing bacteria. And on the right, we have supposedly a wound healing capacity of this tissue. So notice that the cells are able to close this gap, even though they don't start at a high pressure in the beginning. So it's not hydrostatic pressure bringing the cells to the center. It's actually migration, collective migration.
Is this diffusion of cells?
Mm,
Because each one is like moving around kind of at random. So
Mhm.
the random movement leads to the spreading from high concentration to low concentration.
it's hard to tell that just from the video, but we will be able to distinguish both today, I hope, if we have enough time. Because with collective migration, with the cells co-aligning their migration direction, it's not diffusion. Because then they co-align, and then there is a preferential axis because you have this stripe in the middle, and then they close the gap efficiently.
They're moving, which is
Mostly, yeah. I'm not completely sure that this particular demo of the video we are seeing is exactly the implementation we'll do today. So I can't guarantee, but that's more or less what you would expect. But it's hard to distinguish between only diffusion or collective migration just from the video. Running the simulations, it's gonna be easy. So we will review last time. We talked about chemotaxis mostly, and today we'll develop 3 or 4 demos depending on our time. So first of all, angiogenesis. This is the easiest one to build. However, maybe a challenging one to understand the mechanisms. So I will spend a little more time explaining how it works. And then we'll develop the bacterium macrophage simulation without any instructions. So by this time, we should be able to have an objective, like a draw of what our simulation should look like, and then we try to reproduce that video without instructions, just from our modeling ability that we developed so far in this course. Then persistent cell movement, which is necessary for the wound healing simulation. And then finally, collective migration in the wound healing. Maybe I will also cover chemokinesis, because chemokinesis is another way that cells can respond to chemical fields other than chemotaxis. So chemotaxis is spatially sensing of the chemical field, while chemokinesis it's a temporal sensing of the chemical field, and they lead to different behaviors, different cell behaviors, and the mechanisms are completely different. So last time we built the self-constructed gradient. We used uptake of the nutrient field by the cells and a slow recovery rate or no recovery rate at all of this nutrient field. So the cell leaves a trail of nutrient depletion behind. And since it's chemotaxing towards the nutrient gradient, it's as if it's trying to run away from its own tail. So this produces persistent movement, and the cell, as you can see in the picture, in the snapshots, it tries to avoid the trail it has already gone to. We talked about how to specify chemotaxis, and the lambda chemotaxis is basically the strength of the force applied to the cell contours. You can specify that both in XML and in Python using this syntax. We saw that all right. That was received a message about caption. That's okay. Oh, I forgot to say one thing. This section is being recorded, live streamed, and distributed. And my name is Pedro Sensi dal Castel. And this is lecture 11, Chemotaxis, Angiogenesis, Bacterium and Macrophage, Explicit Forces, Persistent Movement, and Wound Healing. So let's go back here. We saw that as you apply more and more force to the cell in cellular Potts model, the speed of the cell doesn't saturates. So at a certain point, you apply more and more force, your cell doesn't move faster, and you can actually erase yourself from the simulation if you apply too much force, because the pixels will try to move towards the gradient by themselves, and it's a mess. Okay. Whenever we are talking about chemotaxis, we have to ask ourselves the specifics of how it happens. For example, where is the signaling molecule? The signaling molecule can be in the substrate, can be diluted in the medium, can be attached to ECM fibers. And how does the cell, for example, sense this molecule? Does it endocytose the molecule? Does the molecule attach to a cell receptor? And as the cell moves, does the cell movement affect the chemical field around? So all these questions are relevant to ask when you build a simulation of chemotaxis, and it can become super complex, even a quote simple demo of cell chemotaxis. How does the cell transduce the signal? So for example, you could do what we've done last class, which is the gradient, the signal gradient applies a force directly to the cell membrane, but you could also model how the signal is transduced and transformed into a force signal using ODE models, for example. We also have to ask some questions. For example, how does the cell move in the absence of the signal? In the simulation we developed, if we remove the signal, the cell doesn't loses its persistent movement. So it's a cell sitting still with some diffusion, and then you give the gradient to the cell, and then it starts moving persistently. But you could have a cell that even without the field, the gradient, it can move persistently and randomly, and then you apply the gradient, and then this cell, which already had the capacity to migrate, now has a bias and anisotropy to the movement. If you're interested in chemotaxis, please take a look and think about these questions one by one. So we're still developing our chemotaxis knowledge and how cells respond to fields and how it can impact a macro scale system. And to study this better, we will develop today the angiogenesis simulation. And I will make a quick explanation of what it is. So there is this cell, and the endothelial cell, it has the capacity to aggregate with other endothelial cells, and they communicate via a diffusible field called VEGF, VEGF. These cells can aggregate, they can signal distant cells, they undergo chemotaxis. At least that's the assumptions we are using to build this simulation today. And they arrange themselves in this, let's say, this net, this grid, this amorphous grid, which we call capillaries. The distance between between capillaries is fulfills a requirement, let's say, of a given tissue, which is providing nutrients and oxygen to all regions of the tissue without transforming the whole tissue into a blood vessel. So you spread uniformly your blood vessels, your capillaries throughout the tissue, so you can provide nutrients to all regions of the tissue with the minimum capillaries possible. There are two types of capillary formation. Angiogenesis, you already have a blood vessel, and then new blood vessels sprout from the previous one. And vasculogenesis is the spontaneous formation of capillaries from the endothelial cells by themselves. So this is angiogenesis in zebrafish. This is a very cool video. You are seeing a thick initial blood vessel, and then it sprouts regularly with a regular distance between each sprout. And you can see, yeah, the formation of the new blood vessels, and you can see that they branch and they connect. And we, for today, we'll assume that this spatial patterning and this communication between the cells is mediated by VEGF. This will be the formation of the capillaries from the endothelial cells by themselves. So this is one of the coolest videos I've ever seen in my life. So the endothelial cells they are seated randomly or almost randomly in a dish, and they are able to communicate, migrate, and form this grid. And that's the simulation we are going to build today. How can, but only by using chemotaxis, how can we achieve this kind of pattern here? Where you start with isolated cells, and they can rearrange spatially, producing these filaments where the filaments interconnect. So the observations are that the endothelial cell migrate independently. They keep a round shape until they reach a range of interaction with other cells to form a cohesive tissue. And then once they reach this cohesive tissue, they stop migrating, stop undergoing chemotaxis. Eventually, they form this continuous multicellular network, and the endothelial cells stay on top of the substrate, so we can model this as a 2D simulation. but it's easy to extend to 3D as well. There are some questions. We will probably address those after we develop the simulation. For example, the cell density in the dish, how does it affect the tissue configuration, the adhesion between the cells, the field decay, which means the range of interaction between cells, because if the field decays slower, VEGF can reach longer distances and the cells can communicate over longer distances. However, if the decay is lower and this field is very diffuse throughout space, you have a lower gradient, and then the cells may not respond so well to the gradient, or you have lower chemotaxis force. So how all those things play out, we'll explore in the simulation. And finding the parameters for the angiogenesis simulation is tricky. I think for today, we will give the parameters, but it can be a real challenge to achieve the patterns. So in mathematics, they model vasculogenesis using various types of models. And here we'll focus on the chemotaxis hypothesis. There are other hypotheses that don't consider chemotaxis or consider chemotaxis and other mechanisms. Here from Gamba and colleagues, 2003, they considered only chemotaxis and the secretion of the VEGF field. And these are the equations of the model. I like this very much because it's very simple. It doesn't look simple, but I will help you understand. So the first equation is basically a continuity equation. It's a conservation of mass equation. It means that you don't have cell death nor cell proliferation. This is how you write conservation of mass. This basically means that the rate at which the cell density changes is basically the amount of cells going out minus the amount of cells going in. And there is no extra term. If you want a cell proliferation, you would make this equal, for example, K times N. And if you had cell death, you could do minus K times N, for example. In the second equation, this is similar to the Navier-Stokes equation. This is the cell velocity field, and it's proportional to the gradient of C, where C is our VEGF gradient. So this equation is basically saying that our cells move towards the gradient. And the field C obeys diffusion, secretion. Alpha is a constant times N means that all cells are secreting C. And it also decays at a rate of tau. So equation 3 we should be familiar with. Equation 1 and 2 we are not familiar with, but the idea is very simple. The one is basically mass conservation, and the second one is Navier-Stokes, where cells move according to the gradient. They successfully achieved the patterns, and this is what we want to replicate using CompuCell, an agent-based model. So this is not an agent-based model. The cells are fields, continuous fields, but it is spatial. So by treating cells as individuals, using CompuCell, we also should be able to replicate these parameters, and we can. So this model cannot track cell movement. In our model, we could track cell movement and see how exactly they move in space. An important consideration here in this model is contact inhibition. And contact inhibition is basically once a cell connects with the tissue, an endothelial cell connects with the tissue, the adhesions between the cells inhibit chemotaxis. So once they form a filament, the cells in the filament, they stop pushing one another inside this blob as if they were still migrating. So once they form the filament, they stop chemotaxis. So it is possible to model that very easily in CompuCell. We call that contact inhibition. So you allow the cells to undergo chemotaxis on medium, but not over other cells. And we'll show you how to specify in XML. Our model structure is fairly simple. We have endothelial cells, the VEGF field, the medium, and we are not representing the substrate, but since our simulation is 2D, substrate is implicit. Our behaviors are random motility. In our case, the cells just diffuse. They don't have persistent migration. The cells have volume. They can have elongation. We can introduce elongation in the model. That would be more realistic. But I think for today, we will keep the cells round, and the cells can adhere to each other. VEGF can diffuse and decay and be secreted by endothelial cells. Interactions. Endothelial cells secrete VEGF, and endothelial cells also chemotax towards VEGF gradients. Endothelial cells can adhere to each other or could repel too. It would be weird, but they could repel. We can try that. And once they come together, they stop chemotaxing over one another. For the dynamics, of course, we're using CompuCell 3D, GGH model, and the diffusion equation for VEGF field. This slide here, we of course we can spend half an hour discussing it, but what basically you can take away from this is that H represents the interactions of the cells. So for example, this first term is the adhesion between cells. The second term is chemotaxis. And the other terms are volume and surface energies. So we should not be too frightened by these equations. And if you have curiosity of what exactly they're doing, we can, of course, discuss that by the end of the class. And the diffusion equation, the field equation, here we have diffusion, which is this triangle, this down triangle squared. This is when you have this down triangle squared and the time derivative, this means diffusion always. I mean, not always, but in this case, in real space. Minus gamma times C, this is the decay, and plus S times delta of sigma, comma, EC. What is that, Emma? S times that, what will delta be?
In this context, I don't know. I mean, I think partial derivative, but doesn't look like that.
It is a Kronecker delta.
No.
Let me help you. If sigma is EC, delta is 1. Yeah. I called it Kronecker delta, I'm sorry. The mathy term.
Okay, so
If sigma is EC, which EC is endothelial cell, right? If sigma is EC, delta is 1, otherwise delta is 0. So what is that?
there, I'm having trouble physically perceiving. So it seems like we're putting in tau is a two-place function, like this. Okay. Sorry, I know people can't see, but I'm just confused, so. It's the wrong way to write tau. Tau is taking sigma, what is it, I or and is this correct?
Oh, wait.
Now I'm
So sorry that there is a mistake here. I think this parenthesis over here should be here.
okay.
Sigma is the cell at position I.
I prime our coordinates in the left.
Yes, I is a coordinate.
So yeah, that makes sense.
Yeah, X and Y.
Okay. And then sigma is just get cell at place.
Sigma, you get the cell at the position. Yeah, exactly.
So coordinates the cells, and then sorry, this is
Let's take a look with the people.
EC is just another cell. It's an endothelial cell.
EC is the type endothelial cell.
Okay, so tau is comparing type of inputted cell to inputted type. Like
Yes.
is this okay? This is a cell type.
So the your rightmost parenthesis is encompassing just sigma, not EC. Yeah. So the type of the cell at position. Yeah. Yeah. I think it's a mistake in the slide.
It's supposed to output a 1 if they're the same and a 0 if they're not the same.
Yeah.
Okay. Thank you.
So do you know what it represents?
Whether the cell at that location is an endothelial cell or is it something else?
This is what delta is doing. So if the cell at that location is an endothelial cell, delta is 1. And then it multiplies by a constant, S, and you sum that to everything else, which means you're adding to the field. Exactly. So you don't need to know what S signifies. It's just a constant. So physically, what does this represent? What are the endothelial cells doing when I write this?
So the endothelial cells, number one, secrete VEGF, and then they also cause other endothelial cells to preferentially adhere to them, right?
Okay, yeah. Adhesion.
So that's the partial derivative of the concentration.
Yes, C is the concentration. Yeah. Secretion. Yes, that's it. Yeah. So at every point in space where you have an endothelial cell, where that pixel belongs to an endothelial cell, you add to the field. So yeah. So the endothelial cells, they are producing this field. Did you understand, Alex and Santu? Did you guys understand what is going on here?
We understood that it's time to get coffee.
What about you, Alex?
I think well enough.
Well enough? All right. Santu, are you following?
Yeah, I'm trying to.
Okay. So basically what this equation is saying, it is that this is how you would formalize these concepts in math. We will not work with these equations, they are under the hood, but basically this is diffusion, the first term, the second is decay, and then every point in space where we have endothelial cells, those points in space will produce this concentration field. Okay, let's keep going. I want to talk a little bit about the contact inhibition in this slide. So these cells, they are chemotaxing. Once they reach contact with another endothelial cell and they keep pulling themselves together, you get those big blobs in the left. If once they reach one another and they stop pulling themselves, pushing themselves together, they only form the filament. Because this long filament, you don't have this, let's say you have a vertical filament. If the cells keep undergoing chemotaxis towards one another, this filament will feel this force, this vertical force, pulling all the cells to the center. So that's more or less the idea. And this is happening biologically because once cells form the adhesions, they don't migrate, at least not in that junction surface.
Do you similar to in the cell sorting simulations, what was happening to the cells that ended up being the outermost layer?
So if Emma is asking if this is similar to what was happening in the cell sorting simulation, where some cells ended up in the outermost layer.
The cells that formed the layers, they were to some extent repelled by each other of the same type. So they wanted to form a line to minimize contact between cells of that same type. I don't know if that makes sense.
I think if you generalize both interactions, the adhesion interaction and the chemotaxis, when the cells are all secreting the same field and following that gradient, yes. If you generalize both concepts, you arrive at the conclusion that you can use fields to make cells repel each other. You can use fields to make cells attract each other. So yes, you are correct in some extent, but you have to abstract the two concepts. Good question. So let's say we build correctly our angiogenesis simulation and we start with a big blob. What we want in this big blob is that it's unstable and it starts sprouting blood vessels. That's what we want. But how does chemotaxis helps? How does chemotaxis helps in this to achieve the sprouting? So in this image here, this point in the snapshot that is closer to the center of the blob, the cells that are closer to the center of the blob, you have a steeper gradient. I hope this is kind of natural because outside of the blob we have zero field. On the innermost parts of the cells we have maximum. So at this point closest to the center, we should have the steepest gradient. And at these points further away from the center, we should have a lower gradient. And what are you laughing about, Emma?
Was, I imagine, like a big arena full of pigs, and you take like pig food that pigs like, and you just like dump a lot of it in the center, and they all rush in, and then some of the pigs get forced to the edges of the group of pigs because they're all trying to get to the food in the center of the arena.
Oh, interesting. So Emma is drawing a comparison with a herd of pigs. A crowd of pigs where the crowd of pigs is trying to reach the food in the center, and then some pigs, the weaker pigs, are pushed out. So since I'm from physics, my comparison is with a water balloon. and someone is squeezing the water balloon. What happens when you squeeze the water balloon? You create those, um, uh, you know, you create those like
Yeah,
this.
if it's full.
It depends on the resistance of, the material. And that's very similar to what is happening here. In the steeper gradient parts, you have a stronger force than on the points you have uh, further away. So all the points are feeling a force inwards, but the points, the innermost points, feel a stronger force. So it's like you are squeezing a water balloon, and then those points, those outermost points, they are extending out. So that's the idea. And as they move in, the gradient gets steeper and steeper, so you have a positive feedback between all those interactions of chemotaxis, field decay, and secretion. So I hope that is clear for you, and understanding this is key to find the parameters in the simulation. This video doesn't work, unfortunately. I have to figure it out someday. So this is our exercise, and the way I planned the class today is that each one of you will help me building each of the exercises. Of course, participation is important in the class, but if you feel if you don't feel good doing it, just tell me and I can do it myself. So for this first exercise, I think this this exercise is basically XML. We don't need Python at all. So it's a good warm-up for the other exercises. And to not be unfair with people, I would like to have one building each of one and not having one person building multiple exercises. If that is going to happen, I will build the exercise myself. For the first exercise, can I ask Sentu to build a model with me? And in this case, I would ask to share the screen, and I can go over you building, go over the simulation with you, the model building part, and then we build that together. What do you think?
Okay, yeah, I'll do that.
Perfect. So let's get started. If you guys want to build a simulation together with us, feel free. Or if you just want to watch and then the simulation file is already on the email and you don't care about building the model because you know you're going to build a model in the other exercises, that's okay. So let me stop my screen share. And Santo, you should be able to
Yeah.
that work with camels too, but
Thanks, well.
I think well enough. Yeah, good job. I also think I think well enough, but I'm not sure. Okay, perfect. So new project. Call it angiogenesis, for example. That would be a good name. Angiogenesis.
He looks confused. I think that he doesn't remember what was in the chat and how I took it out of context.
That's all okay. So boundary conditions, for boundary conditions, I would like that to periodic. Periodic. Yeah. And I want to make a no. No. Where are you, Zoom? I'm messing up everything. Oh, I cannot share like split my screen with Zoom. Okay, that's annoying. So I will have to Alt Tab repeatedly. That's okay. Okay, next.
My Reddit might crash or might work, so
Oh,
I can't
oh,
tell.
wait. Z cannot be periodic because our simulation is 2D. And that's why. Y, this one is Y, that one is
Ah,
periodic. Yeah, it's a little bit confusing. Okay?
If you
Ah,
make Z periodic,
it crashes. Uh,
cells pushing on themselves from below. It's kind of weird.
yeah, yeah, it's very weird. Okay, next. Here, type EC, cell type EC, and add EC for endothelial cell. Okay, add. Next. Chemical fields, we want a VEGF field, VEGF. And add. You have to click add. Okay, next. Here we want, what do we want here? Contact. In addition, we want contact. We want volume. Yeah. Chemotaxis and we could use secretion, but when since we have a field, we can specify secretion inside the field specification. So we actually don't need secretion. Perfect. And let me confirm here. That's it. Next. Here we want a lambda like here we specified lambda 500. And click on that chemotax towards. So in that drop box, we have selected medium and add entry. And when we specify the chemotax towards medium, it means that our endothelial cells can chemotax over medium cell. Let's say medium is a cell. It can chemotax over medium, but it cannot chemotax over other endothelial cells. So this is our contact inhibition mechanism. All right, next. Finish. What we have here, let's try to go through it. I will ignore the comments, but I think the comments are interesting because I try to explain the mechanisms while I set the simulation up. So here we have a smaller lattice of 128 by 128, EC cell, volume, and I also have surface, cell surface, because we had a purpose with this simulation. We wanted the cells more round. So I added the surface constraint. In contact interaction, I set, what is this? Alex, what happens when I set EC to EC contact energy double the contact energy between medium and EC?
That forced them apart. Yeah, and you're also not screen sharing too.
Oh, that's a problem. All right, thank you.
That
Okay.
want to be further apart. You set the contact energy to 20 instead of 10.
But this is a sweet spot. Exactly double. Why, Alex?
Is it enough that you have like some contact, but they don't bunch up together.
No. So the
Oh.
reason is, let's say I have an endothelial cell here and another endothelial cell here, and in between I have medium. Okay? Let's say this is just 1 pixel and this is just 1 pixel. Everything is 1 pixel, okay? So every boundary I have between cell and medium, I have a 10, a J of 10, okay? Because the contact between both is 10. So when the cells are separated from each other, I have how many surfaces, how many contacts with medium? I have this one and this one. So I have 2 times 10.
Oh.
I have 20. And when the cells are on direct contact to each other, I have only one surface. So if I want to make this configuration as likely as this configuration, I have to set this energy to 20 and this energy to 10. So this means neutral. This means that the cells don't have preference for being in contact or not. All right. Let's keep going. Here I set, of course, the parameters are not exactly the same as in the slides because this is my implementation for another purpose, not for not for the class purpose. And here I set a lambda of 2000, okay, and with a saturation coefficient. So this is not a linear response. This is another kind of response. And to compensate for the chemotaxis expression, I had to increase the lambda here. I also had length constraints, but here the lambda length is 0, so it doesn't matter. It's as if I didn't have this plugin at all. In my diffusion solver FE, I have my VEGF field. It diffuses with diffusion 1. It decays, it diffuses inside cells, and it doesn't decay inside cells. That's actually important because if it decays inside cells, let me go back to the slides. If it decays inside cells, this big blob would not have this huge red, this. But I think it could work. I mean, it's not, maybe it's not super important. But you will have steeper gradient at the borders if you do that. My EC cells secrete at this rate, 0.1. So it's a constant secretion rate, not a constant value. And of course, periodic boundaries. And I initialize my cells evenly spread throughout space. So in my uniform initializer, steppable, I have a gap between the cells, and I seed 4x4 cells throughout space. And when I run this, let's see what happens. Of course, this simulation already has the right parameters. So it's not a huge surprise that it works, but it can take a while to figure out the right parameters, even for someone relatively experienced with CompuCell like me. And I already built this simulation several times. It could take me 10 minutes to figure out the parameters that make this simulation work. What is interesting here is that you can see that sprouts never stop happening. The cells, the tissue always creates new sprouts and the sprouts grow. So it's this system never reaches equilibrium. It's always out of equilibrium. And this is This is due to the instability of the field and chemotaxis. You also can note that these small holes in the tissue, they tend to close. And then because the gradient is it's less steep than on the outer holes. Because in the bigger holes, because the bigger holes you have more space to decay. In the smaller holes, you have less space to decay. So the cells tend to close these holes. And when they close the holes, you have a high density region of cells, and then they sprout new capillaries. So this system is highly unstable, and that's one of the reasons it's hard to analyze. So people are still figuring out metrics to analyze this type of system here. You see this hole just closed, and then we have a high dense region, and the simulation stopped by default. Yeah, very cool. So I have the simulation. The simulation is done. You have access to the simulation, and since you have access to the simulation, I will distribute these questions to you. I will answer the easiest ones myself, and I will leave the interesting ones to you. So let's see. I will answer the last two ones, and I will leave the first question to Emma, the second question to Alex, and the third question to Santhu. I think that's fair. So first, Emma will answer, the second one, Alex will answer, and the third one, I'll have Santhu answering. Santhu, were you able to run the simulation from the...
Yeah, I did run the simulation.
Good. So you guys can run the simulation and try out in the simulation and see what happens when you change the specific parameters that the questions are referring to, and then answer your question based on that. But before you run the simulations, guess what will happen from the mechanism that hopefully you all understood. So, well, if I understood the mechanism correctly, I think if I increase the number of cells, this will happen. And then you run the simulation, see if it agrees with what you expected, and then we'll discuss about it.
So I'm trying to open, what I know after a meeting with Maciek is that on Mac, the diffusion solver we're using is not very good. It actually takes a very long time. So he suggests that I instead use this one. But I think that in this case,
That is not, yeah.
so if I had a diffusion constant of maybe 30, I think it would be fine. But with 1.0, I think it's enough to basically wreck the simulation. Because I noticed that, I don't know, I just had the feeling that the diffusion would not place number. So I mean, and you know, I'm trying to play with, I'm also confused by this. One of the things that I tried was just changing this to be lower. And look what I see. So it more quickly forms those filaments, but they don't become thinner and thinner. Like they don't get to one cell per filament width. See? And I think the problem is diffusion, but I'm not.
Okay, let's try something then. So Emma is having issues with her PC, so she will take my place and do it on my simulation.
Okay.
Come here, Emma. So it's already time, and so this is the simulation right here. So please take a seat and let's find out what is going on. So this is the basic simulation without anything. Yeah, run it.
That's what you meant by density.
Oh, what have you done?
So I didn't have any space between the cells in my uniform initializer at the beginning.
Yeah.
So I thought density was actually having to do with volume. I was a bit confused about what it meant.
Oh,
Yeah, I was okay.
yeah. I think this is the default settings,
Okay.
isn't it?
I'm
Okay.
not sure what I have.
Yeah.
I can't remember, but I don't remember changing anything. Let's see. So that
Okay.
would be in the uniform initializer here.
You have to reinitialize the
Right.
yeah. Yeah, so here it's not gonna work because it's already initialized. You have to change the
see.
XML.
Okay.
Yeah.
There. Here. Okay, the gap.
Gap.
Let's make it 0 and see what happens.
Oh, 0 it's not interesting.
So it's no gap.
Yeah, there's no gap.
Yeah.
You'll never have angiogenesis. You don't have medium at all.
Huh. Wait, now I'm a bit confused.
If you make gap 0,
So there's going to be a square-shaped region of cells in the middle of the medium. But that's like what that was just to demonstrate. I mean, okay, what we saw earlier in the slide.
Save and run.
Okay.
Let's see.
I'm really lost here. Wait, it would be
Alt
here.
tab. Yeah, yes.
Okay.
We can do that. Alt tab is the quick. Yeah.
Oh, but now you have
Yeah,
the whole
it's-
field is okay. Yeah, I see why you said there was no medium. But what if we just make the region smaller, like this?
No, instead of setting gap to 0, you can set to 2 or 3.
Right. But
Instead
like,
of-
I mean, what if we have like a region of cells in the middle surrounded by the medium on the edges?
You can do that too, yes.
So that's what I
But
expected.
yeah, but you are not sure if it's the same amount of cell. I mean, not the same amount of cell, but
I see. Okay.
okay, that's a very, very
Yeah.
dense.
So there's
Run,
there's
let's
less
see.
space for them to make. Yeah. Okay. So, I mean, there's just not that much space for there to be holes between the cells.
Mhm.
So you're forcing them together anyways.
Yeah.
Okay. Now let's make it. We already tried 5 at the beginning. So let's
Mhm.
try 8. So yeah, it will be, but at a certain point there could be long filaments. It'll take them longer to come into contact too.
Mm-hmm.
So before it was actually empty space that was the limited resource. Oh, it's so fast.
Yeah.
It's pretty fun when it's really fast.
Yeah, it
So
is.
the holes will still
Mhm.
be contracted together
And
and there'll
sprout.
be more growth out.
Yeah.
Yeah.
They sprout new filaments.
Yeah.
So
So maybe if it's 50-50, it's the most interesting.
can you create a mesh with this low density? A network?
No.
No.
Now the cells are the limited resource.
Yeah.
So you want neither one to be the limited resource, which maybe this is what we get here. Okay. Yeah, so that seems pretty balanced.
Mm-hmm.
Maybe a little bit lower.
Mm-hmm.
Yeah. Okay.
Okay.
Cool.
That's good. Thank you, Emma.
Mhm.
So I will ask to everyone in person in class to applaud. Applaud? Applaud. I'm not sure if that's the correct word. Yes. So yeah. Applauding Emma for the awesome contribution. And now let's have Alex answer his question. Alex, how does cell adhesion affect tissue configuration? And hopefully you will answer that and show it with your simulation. And maybe my Zoom crashed.
Still seems to be working.
Am I screen sharing?
Yeah, we see your PowerPoint or the PowerPoint software, not the presentation.
Oh no. Do you guys still hear me?
Yeah.
my Zoom is not responding. I will click
No.
wait for the program to respond and hope for the best. Hayden, are you there?
Yeah.
Are you co-host?
No, but you can make me. Or well, I guess not
I
if your Zoom's
can't.
not responding.
I can't. My Zoom crashed. I have to kill it.
I think James just took back over the host, so he can give me and you.
Okay, so try to make contact with Mr. James. Yeah, it just crashed. I mean, what is going on here? Yeah. Wait for the program to respond. Oh, well, um, I'll try to kill it, and I think is James in the call in Zoom?
Yeah, I just was
Oh, okay.
closing the screen share so you could try again.
Okay, I will kill Zoom at my end and re-enter.
It looks like you're okay. It says started screen sharing, so
All right. Um,
your
and
video
There's a
feed is working all right.
there's a clone of you in the room now, though, a frozen clone.
let's kick him. Let's kill him. Who is this Pedro? Yeah, can you delete him from the Zoom? Okay, good. Okay,
Okay, give me a
okay,
chance.
perfect. Go on, Alex. First, tell us what you thought. What was your thought about the question, and explain what you found.
So my thought was going to be that if you set the contact energy too low, they're just going to aggregate together because there's a way to minimize energy would be to come together and not a blob as I originally had thought, but they'll just come together. You set it too high, and they're never going to want to come together. So either way, I mean, on the one end, you'd have sort of the same structure, but larger. The other end, you just have no structure at all.
Mm. So you would say that a very high contact energy between endothelial cells would not kill the pattern?
It eventually it did. So yes, eventually, but it takes a little. It took a while. I had to double it.
Okay. Can you show that to us?
Yeah. This is the original one. That takes.
Uh-huh.
Then if I turn it to 40,
Mm-hmm.
about they'll still come together, but they're maybe it was that they would come together in like little clumps, but they would never. They
But
might only.
you can see more or less that they are trying, right? They are
Yeah.
trying to form the filaments. And as you decrease the energy, at a certain point, the adhesion will win this hydrostatic effect, right? This hydrostatic force. and eventually it will dominate. It is possible to find the sweet spot, but we can see that we don't have the same structure as before, right? Here they seem to aggregate more. We don't see sprouting. Or we do see sprouting, but the filaments are thicker. So it's a bit confusing sometimes, because let's say you take this simulation with the thicker filaments and then you zoom out. And then you find that the structure is the same, but you just zoomed out. You know? So this is one of the reasons it's not so easy to analyze this kind of simulation. Can you see that, Emma?
Actually got your code working.
Oh, good. Running good.
Yes, but there are limits.
Okay. So, at last, Sentu, what are your thoughts on how does field decay in medium affect tissue configuration? What did you think? And show
So
us.
I've tried with the decay constants from 0.1 to 0.3. For the lower decay constants of 0.1, I found the filaments to be bit stronger and thick
Mhm.
because they have like more stronger attraction towards each other compared to the higher decay constants of 0.3. I'll just screen share it. So this is how it kind of looked with the decay constant of 0.1.
Okay, good.
Yeah.
And what happens when you increase a lot the decay constant? So let's say 0.5.
There is much reduced interactions between the cells. Like it exhibits a like uh longer and thinner filaments, and there are like big cell blobs, and they're like kind of like less aggregating to it
Mhm.
because of the weak attraction forces, I guess.
Yeah, so but the mechanism behind it is the other way around. Here you have a lower decay. With a lower decay, you have a less steep gradient in the border. If you have a less steep gradient in the border, you have less force acting over the filament. If you have less force in the filament can be thicker. On the other way, if you increase the decay, the gradient is steeper in the border, and then this thick filament is squished and elongated. So it's
Oh,
the other
okay.
way
Oh,
around.
it's the other way. Okay.
Yeah. But that's good. Thank you.
Thank you.
Okay, I will now screen share the presentation. How can we predict angiogenesis from a parameter setup? We can discuss about that, but I think we don't have an answer. Maybe AI. I think people are still working on that. How can we guess what parameters are in a real angiogenesis using the simulation? We can try, for example, to match the pattern, to match the real pattern using the simulation. The problem is you can match the pattern for many many parameter sets. So it's a very hard question. So those two questions, they maybe I think there are still open questions. There are other exercises, for example, removing contact inhibition. I can do that here quickly. Yeah, I closed my to edit because I thought it was interfering with Zoom. No problem. So this is the original simulation. Chemotaxis towards, if I delete this, oh, it does work. Interesting. So I removed the contact inhibition. Now the cells in the tip of the filaments, they sense they can undergo chemotaxis over the other cells, over the neighboring cells in the tissue, and then we just get these big blobs.
You're not sharing the simulation, just the
Oh.
exercise. So you better run it again.
Oh, it should. Do you see it now? Do you guys see the simulation?
That's fine, yeah. Why don't you
Okay.
try starting it again so you can see the...
Yeah. Okay. So this is the original simulation. Don't ask me why it changed color. And this is our chemotaxis towards medium, which means the cells can only undergo chemotaxis over medium pixels. If we disable that and they can chemotax over anything, including other endothelial cells, these filaments, they get crushed because all the cells now are compressing its neighbors and we form these big blobs. All right. For different cell densities, we explore that we can also start the simulation instead of having the cells spread out in the simulation. We can start with a big blob in the center that was Emma's idea. So gap 0, but then let's create it, let's say from 40 to 80, from 40 to 80, and we should see sprouting from this blob. Yep. So we can achieve the pattern even if we start with a blob. This is only possible because the system is unstable. It's unstable and it's stochastic, because if we started with a perfect circle and no stochasticity, we would not leave the stable configuration. But we have, like, the blob is wiggling, so you will create bumps, and these bumps will grow, forming filaments, and the valleys will be pushed inwards, pulled inwards. The diffusion length is basically controlled by the decay constant. The diffusion length is, let me think, is the inverse of the square root of the decay. So they're associated. If you want to decrease the diffusion constant by a factor of 4, you sorry. Oh, this is, oh, wait a sec. Diffusion constant, decrease the diffusion. Oh yeah, it also depends on the diffusion constant. It is the square root of d over decay, the diffusion constant over decay. And then we basically explored that with Santhu question, but instead of changing diffusion, we changed decay. All right, now we have a challenge for you. And the challenge is building this simulation. This is also chemotaxis, only chemotaxis. And by now we should be able to look at a video like this and build the simulations ourselves without instructions. The simulation is not in the files. But you are able to build it nonetheless from scratch. So here, what do you need? You need a cell, a bacterium cell that secretes a chemical, and the macrophage that undergoes chemotaxis towards medium or towards anything, but following the gradient of this field that the bacterium secreted. If you get stuck or if you're unable to develop the simulation, instead of building the simulation, you can write down the model structure, how you would implement it in CompuCell. So I will give you, it doesn't need to be this level of detail. You can start without these red cells, these cells that are just standing still, and you can ignore them and have just one macrophage and one bacterium in a smaller lattice to run faster. So the idea here is to simulate one cell pursuing another cell, chasing another cell. Start from the existing demo bacterium macrophage 2D.
Okay.
And you have to modify it to tune the parameters.
Okay. Yeah. I'll do that.
Necessarily changing the colors. Because that's
Do you have your macrophage chasing?
I don't know. Oh my god. It's getting stuck to the red blood cells. I think the macrophage is displaying white. So let me
Yeah, I think it is. Yeah.
Oh, that's why I intended this. The bacterium with these small. Oops.
Make less bacteria, increase lambda chemotaxis to see if they
But you
can
can
find their way to the bacteria.
fewer red blood.
Much less cells.
And then looking at Texas.
Yeah. Let's see what is the number used.
Here.
200.
Oh, 200. That's fine. I didn't remember the values. Okay. Let's see, it's supposed to be secreting for the flame. It's not diffusing very much. Maybe I think that's the market page.
Reduce the lattice to increase probability.
But okay, the signal is it just not diffusing now? Maybe I should make it secrete more at this time.
Yeah, both reasons are good reasons. Yeah, so Emma is trying to tune the field parameters, which is always a work to do when we develop simulations with chemotaxis. And Alex, how are you doing?
Yeah, just trying to fix the number of cells, making the lattice smaller.
Could you make the macrophage chase bacteria at all?
There's just way too many, so it's hard to tell what's going on. Yeah, I think it's doing it. There's just too many cells.
In this simulation, I could. There's a PIF initializer. Oh.
You know, I tried to do this last week and I just couldn't get the chaser to get the scent to play. Yeah, I just don't know. Maybe I have to spend a lot of time tuning with careful.
Using the logarithmic chemotaxis may help.
Oh, I don't really understand what that means.
Yeah. No. Here. So the force applied to the border of the cell is just lambda chemotaxis times the gradient. Okay. The logarithmic chemotaxis, you divide by the concentration of the field. So if you have an exponential field profile and you're further away from the source, and you divide by the concentration, you get a stronger chemotaxis. And then if you are too close, you get a weaker, but the gradient is steeper, so they balance each other out. So you can regulate the sensitivity, the response sensitivity of the cell to the gradient by using logarithmic chemotaxis. And in reality, cells do tune the response to the field levels. So this could do, and you can do that by adding. Yeah, you can do that two ways. You can set lambda in Python, or you can do that in XML. And in XML, let me show you how you will do it. I will come here to the manuals. Python reference manual. Chemotaxis. And yeah, this is the form we want. And to do that, you just add this line. And let me send it. It's in the chat. If you add that, your cells suddenly will adapt to the field level. The scale coefficient is this S, which is basically first of all, if your field is 0, you don't get divergence. And second of all, when your field is very, very low, your response is basically lambda over S. So if S is 1 and the field is low, your chemotaxis, your lambda chemotaxis effectively is lambda if S is 1. And then if your concentration is too high, your effective lambda is 0. So S could be 1. I think 1 is okay. Yeah. That definitely helps in the simulation to make the macrophage, let's say, smell the bacteria from afar. Did it work for you, Alex?
Sort of.
Okay. For the purpose of getting you guys to build a simulation, I think that's good enough. You get a sense of the challenges you face when you build a simulation from scratch, which is basically tuning the parameters to achieve the desired effect. Yeah, that's always a problem. There are model extensions you can try yourself at home. We're not going to do all of those today, maybe none of them. And we finally get to the most important part of this lecture, which is persistent cell migration. So one of the reasons it's hard to make the macrophage reach the cell is that the macrophage, if it doesn't smell the cell from afar, it doesn't move at all. But in reality, macrophages, especially neutrophils, they are always on patrol. There are different types of macrophages. Some don't migrate, some just sit down and wait to be activated. But those kinds of macrophages that we saw in the video, they are always on patrol. And then eventually they meet a bacteria or a pathogen, and then chemotaxis start playing a big role to direct the macrophage directly towards the bacterium. So in persistent cell migration, what happens is instead of solely diffusing, our cells navigate more persistently. So they choose a direction, they migrate towards that direction for a certain period of time, and then they change direction, migrate on that direction again, and so on and so forth. So in the long scheme of things, the movement is diffusive, but if you analyze if you see the cell movement, you will see that it generally migrates in a certain direction, then it makes a turn and migrates in another direction, so on and so forth. So how do we make those macrophages patrol? We use external force plugging. You can implement the external force from the wizard. So in the cell properties and behaviors, you can check this box, external potential local flux. And you have to check center of mass if you want to use the center of mass algorithm for external force. So basically, external force works in two ways. The first one is like chemotaxis. You promote copies of pixels in the border of the cell towards the direction you want. And the other way is the center of mass based. In the center of mass based, you accept the copies that make the center of mass of the cell move in the direction that you want. And then you specify the direction that you want the cell to move. So I will demonstrate that. Yeah, I will demonstrate this plugin here on my own. So let me create a new simulation from scratch. Whatever. Cell, no fields, contact volume, external potential, local flex. That's generally what you want to do, not the external potential. The external potential you cannot change in the Python. And then center of mass, it's already checked. Okay. Now let me close these ones. In this new simulation, we have the external potential plugin, and we can specify these lambdas here. I'm not sure about the syntax. Let me potential, external potential plugin. Okay, this is the syntax. External potential, specific cell types. No, let me use the simple one. So here I'm basically specifying a vector. Let me do 10 minus 10. And let me run this. So all my cells are moving to the right. And I have a question for Alex. Why are my cells moving to the right if my external potential vector is minus 10 on the x-coordinate? So it's pointing to the left. Why are they moving to the right?
Kind of like the normal force, like they're just going to do the opposite of what the. Am I thinking of it wrong?
Yes, you're thinking it wrong, Alex. So the reason is because in CompuCell, things minimize energy. So this is the direction where energy would increase. So the cells are moving the other way. All right?
Just going periodic.
Yes.
They're spreading out.
Mhm. The other algorithm is the center of mass based. And this one, if I run, you see that the cells don't move at all. Why is that? Why aren't they moving? Center of mass based. I sent in the chat. So, Santhu, why are my cells not moving in the center of mass based algorithm?
Maybe there are no like external forces acting.
Well, in fact, they are moving, but they are moving slowly. If I set maybe 100, yeah, now it's clear that they're moving. So the reason is when you apply the force to the cell borders, you are directly intervening in the copy attempts. So the effect is much more expressive. Where if you wait for the cell to make a copy and then see if the center of mass moved in the direction you wanted, and therefore the potential would the energy would decrease a little bit, and then you accept or not that copy, the effect is much milder.
You know, I let mine run and they started spreading out.
Why are they doing this, Emma?
They're all trying to get to the front and with, because then they can move slightly faster than where they in the back. Because if they're in the
Because they are not interacting, they're not being like bumping into other cells. Interesting. Mhm. Interesting. Yeah, so Emma was running a big blob of cells migrating to the right, and they formed a weird triangular shape. And then she was trying to figure out why. She came up with an interesting hypothesis, but we are unable to prove it. Interesting. But to make one cell move persistently, and this is our next exercise, I will have Emma do it for us. And since Emma has a problem with her computer, she will take. What are the limits?
If I put the diffusion constant equal to more than about 3, it is really quite slow. But you know, below that,
Oh yeah,
it's
for everybody it's slow if you put a greater than 3. Yeah, for everybody. Okay. Yeah. Okay. Maybe that was our fault to not explain that in class.
my fault explained it, but he made it seem like it was something with mag. So now
Ah.
it might be worse.
Okay, so Emma, is it possible for you to share your screen? I am willing to wait. And while Emma opens the Zoom to share her screen, I will explain what is. Oh, I have to screen share again because I will explain the idea behind using our external potential plugin to make cells move persistently. So first, we define an angle for each cell in the simulation. And we'll define a force with the lambda parameters following this angle, cosine of the angle in the x direction and the sine of the angle in the y direction. And then we'll add a random number to this angle. And then this angle will vary a little bit. As the angle varies, the cell changes direction. Is this idea clear? So we assign an angle to each cell. We tell the cell to migrate in that direction. And each step, we update this angle with a random fluctuation. Is that all right? It's super simple. Why does that produce persistent migration? Because if you add a small random contribution to the previous angle, you are more or less moving to the same angle. But in time, these random contributions add up, and eventually you're moving to a direction completely different from the initial one. They are not doing a random walk because in a random walk, every step is completely independent from the previous step. If we are doing this, the next step is super correlated with the previous step because we are like adding a little bit of noise to the previous step. It's like inertia. Perfect. Yeah. Good insights. So please, Emma, go ahead, share screen. We'll build that together.
with the folders. I don't know.
Yeah.
Oh wow, this is very strange. Okay, I want the toilet. Oh, Zoom, why? Okay.
Can you unmute so people can?
I'm unmuted.
You can unmute. Huh? Yeah.
Hello?
Okay, so Emma muted, and I will follow along with Emma. So, okay. First of all, you have your external plugin, external potential plugin. Change that to center of mass based.
Oh, it's here.
Yeah, it's already there.
Is it like this?
Yeah, no. Off, yeah. Perfect English. Oh. And then delete line 36. Good. Now, do we have our cells? How are our cells disposed in the lattice?
You mean the initialization?
Yeah, okay, that's good. To make it run faster, let's start with a small lattice, like 50 by 50. Yeah, yes, 50 by 50. And create like four to 6 cells. So from 20 to 50, whatever. No, yeah.
Okay.
And width of 15, let's say. Yeah.
And you want gap of
0, yeah. And then neutral contact energies to make sure if we ever get collective migration, we would prefer to have, yeah, neutral to compare. Okay. Now let's go to your steppables. And let's start by defining initial angles to each one of our cells. And we do that so in the start function, you will write cell.dict. This is the cell dictionary. And you will assign a variable for each of your cells. Okay. So loop over cells in the start function, for cell in cell list by a self cell list, and then create an angle for each of your cells.
Cell.dict is initialized at the beginning, right? I don't need to make a dictionary.
No, it's built in. You always have a dictionary, so you can add, subtract, do anything you like in the steppables, in the step function, wherever you want.
To extract all of those data in the form of a NumPy array, so you can do vectorized computation, it would be a lot faster. Actually, I don't know whether it would be faster. I don't know how NumPy is.
You can definitely create your own NumPy array to do these kind of things. But yeah, if your dictionary is not like ultra big, it's not a problem. Angle.
Okay.
Yeah, just assign a random value. So, and NumPy is already imported. So np.random. This is always funny.
Now they're changing how you make a call for random numbers in NumPy, and I don't understand.
Oosh.
I don't know if you
That's gonna screw my life. And then 2π because, why not? Good.
What was it? X. Okay, directory X.
Direction X.
Okay. Yep, that makes sense. Listen to that.
Yeah. Define a force. We don't need to write propulsive force. Force. or proportion. 20 would be too little. Try 200 because we're using center of mass based.
What's this alpha parameter again? Persistence.
Alpha will be our persistence. How would you implement persistence?
The standard deviation. So the standard deviation would be 1 minus that, or
Yeah, that's a good one.
that's
Yeah.
how I do it in my thing.
Good idea.
Yeah, but for the sample when you vary them. But that would be in the step function. Okay, I don't want to print this already. No, we just want to add, gosh. Is this right? I don't remember.
Yes. Yes.
It's
No, no, you can just do random. It's a number between 0 and 1. And then subtract a half and divide everything by and multiply that by 1 minus alpha.
Okay, yeah.
Or you do random dot uniform between -1 and 1 and multiply by 1 minus alpha.
Yeah.
Single star. Yeah.
Let's see. This is so
That seems no. It's alpha or persistent. Oh, okay. Persistence. Good.
Right, and then we want to update this.
Yeah, we want to update that and we want to pass those values to our lambda force parameter. So to get that, you go to CC3D Python in the menu, yeah. Force, where is force? External potential or something like that. Go to cell attributes. Go to cell manipulation, maybe. Constraints. Lambda motility. No. It's apply force to cell. Sorry. Go Python. Constraints, apply force to cell. Perfect. Okay. Okay, what do you do here?
These. This?
Yes. Yes. Perfect. Not perfect, but almost.
Negative?
Good. What else? Our force. Our force parameter.
Oh, this one?
Yes.
Oh, yeah.
Yeah. Because you see, we have sines and cosines, right? So they are constrained between 0 and 1. And then you modulate by your force parameter, and that's good to go. That should do. Let's see. Run it and let's see what happens. Yeah. You still have another image. You have to kill it.
Wait, I thought guessment stopped, but maybe guessment.
Yeah. Good, nice. You see that they are eventually changing directions or not?
This one's taking this curvy path. Yeah, here, this one just moved.
To test that, let's try lower persistence. And we'll see how sensitive the simulation is to the persistence. Look how low the persistence is by setting it to, by lowering it to like by 20%. You know.
Lower it by 20%?
No, we lowered by 20% more or less, and the persistence reduced much more than that. But when you see the simulation, yeah,
See, if it's 0, they don't get very far. They just cancel
yeah,
themselves out.
they do whatever. This is your diffusive movement, because you always pick a new complete random. Almost, yeah, to be completely random, you would have to multiply by 2 pi in line 34. Yeah, to be completely random. By pi, actually, because it's between -1 and 1. But that's okay. Now that we have that working, I will share my screen again and advance on the slides. And yeah, we have our noise parameter. This is adding plots to the slides. Let's not worry about that. We did that too, which is updating our lambda parameters. We done that, so we have our noise parameter. Okay. And now our next objective is to have collective migration. And the easiest way of doing collective migration with this setup is by having the cells go to the direction they were going in the past. So instead of fully updating out of nowhere the cell migration direction, we update the cell migration direction based on the displacement of the cell in the previous step. And how does that make the cells move collectively? Well, imagine two cells coming together and they bump into each other. After they bump to each other, they stop their movement because they bumped into each other. And then you will feed into the new direction will be associated with these new displacement, which is 0. So they'll stop moving because now and for example, if one cell is doing this and the other cell is doing this and they collide, now this cell will be dragged by this one. Let's say they have a low contact energy. One cell will drag the other. And if you have a positive feedback between cell displacement and cell migration direction, you can make the cells co-align. I was thinking of a better way of explaining what I was trying to explain. So basically, the idea is this. When cells interact, they change their displacement because they bump into each other, they are dragged by each other, they interact with all sorts of stuff. This interaction leads to a change in the cell displacement. And if we feed this displacement to correct the cell migration direction, we can make cells align. We can make cells co-align. Yes. We will work out on an example. Yes, on the board.
well frame two, which is that, well, somehow this is actually not possible. That is not a very good purposes. Okay. These guys just collided, right?
Mm-hmm.
Okay, so what's going on here? This cell is traveling with this velocity vector. This cell is traveling with velocity vector of
Mm-hmm.
straight up.
Mm-hmm.
Then they collide. Okay, so what happens if you do what you recommend to these?
Okay, I'll have to draw on my notebook because I don't have three hands. Yes. Yeah. So I will draw it in the paint so we can see on the screen here. So one cell is coming this way, one cell is coming this way, and suddenly they are in this state, and then they come to this state where they are trying to do this. But now that they are restricting each other's movement, they can only go up and down. Okay? So their displacement, although the current migration movement is crossed like this, their displacement will be up. You will project this, and the displacement, which I will draw in red, is up. And then we take this displacement and we feed to the new migration direction. And then the new migration direction is this and this. So we make a contribution to the cell migration direction.
So if we didn't do this, the cell migration direction would be determined just based on those old black vectors. But
Yeah.
those don't accurately represent what the cells did in the simulation.
Exactly.
So we have to just adjust for the actual physics of the situation by putting in the red vector man.
Good way to put it. So what Emma said is that to achieve collective migration, we have to feed into our migration vector what happened to the simulation physically. So if there was a collision, how can I input the fact that there was a collision into our mechanism?
Is there like some cell manipulation thing that's like get contact surface or something? If it's non-zero, then
It is. You can do that, but and you can ask your neighboring cell what is its migration direction and use that information to feed yours. But the thing here is, by just using the displacement of the cell on the previous step, we can make it work. We can make it work. Because if they come in contact, especially if they have high adhesion, you restrict the displacement, and eventually the migration vectors will align.
but we will have to infer which cells collided with each other based on the previous displacement.
No, you just feed in the displacement. Let's do that. Let's go hands-on. So share your screen again. We're using your simulation. And then, okay. So let's try to do everything. I mean, yeah, I can put the slides. So first, let's get the cell displacement. Here, they are calling velocity because either way it works, because Monte Carlo step is 1. So if you divide by 1, doesn't matter. And you can make it a NumPy array. I know you like NumPy arrays, so we can do that. And basically, we already have built-in variables for current position and previous position, which is cell.xcom. And cell.xomprev with uppercase P.
Can I go back to the screen that I actually have?
This one?
Yeah.
Okay.
So, center mass be a step. I think we'll just do that. That's time step, right?
What?
Oh no, you're just putting in 0 for Z then.
Yeah, 0 is the Z.
Always because we're in B.
Yeah. Let's see.
That should be fine. Why do we need? Okay.
Yeah, because inside your NumPy array we are giving a tuple. That's a tuple, and then you transform the tuple into an array. All right. So you have the displacement. Now we want to normalize the displacement because, well, let me see.
It's just the length drops, right?
Yes. The direction of the displacement. That's all we want. Just the direction of the displacement. Let's try without normalizing, because I think it should work the same way. So you do this. Now you're going to update your X direction and Y direction based on the velocity. Okay, so you'll do cell.dict X and cell.dict dir Y, dir X and dir Y, these two variables. They will be, copy those two lines and paste on 42. Yeah, let's work on one of them only. So in dir X is going to be equal to dir X. Yeah. Times a correction parameter that you can call alignment, for example.
That for each cell or for the simulation?
For simplicity now, let's make it equals 0.99.
And that's not force persistence?
No, they are different things. So you can make force persistence equal to 1. And you think, oh, so if it's equal to 1, they'll never change direction. Well, but if they have an alignment parameter, they can. So let's study that. Actually make it 95. 95 is the sorry, the alignment parameter, which is 99 up there. Make it 95, yeah. I tested that yesterday. Okay. And then it's gonna be this plus Y instead of X, Y in the 44.
But what are we?
Yeah, it's this. So when we multiply it by self-alignment, we are taking just 95% of the previous, and then we are gonna make a correction. So the other 5% are gonna be in the velocity direction. Okay, so it's gonna be 1 minus NP, 1 minus self-alignment. Yeah. Times the velocity on the X direction. Which is cell.dict velocity, and then square brackets 0. And then the analogous for Y. Y1? Yeah. Uh, that should work. Let's run. Oh, no. You have to pass 3940 to after, yeah, after the correction. Yes. Lambda z doesn't matter. Let's see if they can align now. Oh, you see that then that when they come in contact, they change direction a little bit. Let's see if they can align. So we have more kinds of interactions now than before, right? Especially when they collide, they change a little bit their direction. Now, there are two interesting things we can do here. The first one is increase the cell density. The second one is increasing the cell affinity. What do you prefer?
Let's see what happens when we increase the cell density.
All right. So create cells in all space, so from 0 to 50, and give it a 1 gap and with 10 more cells. 1 gap, 10. Yes. Let's see how that goes. Let's see if we find clusters moving together. We can try doing the limiting case, do confluence, full confluence.
This.
0 gap, perfect.
Well, that doesn't say much.
Increase the force. $500. Oh, wait. Go back to the steppables. You are multiplying by 200 in line 43, 44. You should be multiplying by cell dict force. Let's see how it goes now. Okay. And I would expect that if we wait, reduce create more cells. Reduce the width, yeah, to 5. Yeah, 6. That's good. Whatever. Double the cells. Oh, you see? I think are they aligned?
Seems like they're getting there. Yeah.
Yeah, this one is a bit more work. Let's try only like 4 to 6 cells, but then more contact energy, like less contact energy between them.
Yeah, the numbers don't work out, did they? Each cell has width 5, is that right?
Yes,
Okay.
that's good, I think. Yeah. And reduce contact energy to 10. 20 to 10. Yeah. Let's run that and see.
So now they have a preference to stick to one another.
Let's hope. Yeah. And then once they stick, we should see them crawling as
Yeah, I think I do see that.
clusters.
Yeah, there was one. Oh, it's slow. Yeah, here's a cluster that, yeah. Yep.
I mean, maybe, you know. I'm not fully convinced yet. Eventually, we should see just one cluster migrating unidirectionally. One thing we did not do was scaling the velocity. So we can try to do that. Maybe that's super important and we didn't do that. So go back to your steppables and normalize your velocity by the norm of the velocity. So you can do cell.dict and p.linaug dot norm, I think.
Let's see what this is.
Yeah, no, it's not gonna work because you don't have cell velocity defined yet.
Well, we do because we defined it. Oh, I see, but it would be the previous one. Okay.
Yeah,
Well, we'll just have to.
you have to do that separately. Velocity equals velocity over norm of velocity. Yes. Perfect. That should do. And let's try again the same setup. And let's hope the velocity is never zero because we'll have a problem. I am not convinced. So I will screen share instead and show you what I have, which maybe is the same thing. So it's simulations, persistent migration. Let me run this. Look how fast it is. So yeah, it makes just the exploration more interesting.
It's pretty similar to what we saw just a second ago.
Yeah, let me increase the number of cells. That's clearly a cluster migrating together. Clearly. Yeah, maybe that's we did 10.
We did.
10, 10, 10. Yes. Look, they're one cluster migrating persistently, right?
Mhm. Yeah, self-dot alignment.
Let me see if we have something different here. I have okay, this was okay. I have an exception for norm equals to 0, but that's, yes, so that's not important. And then I do this update. Norm, so I just have another variable, who cares? Yeah. Oh, yeah, we are missing this. We have to update the angle based on the cell displacement. Can you share your screen back? That was the,
Do this year. I wasted so many hours of my life coding manually. Yeah,
wow.
I got such a stupid.
All right, let's do it.
Okay, not
Ca-
oh, there it is.
Can you guys hear Emma, by the way?
Mhm.
Yeah.
You want to do one first?
Yeah, why first? Yep. Yeah, doesn't matter. Yes, that's it.
Okay.
Arctanto. Arctanto. It feels super different already, in my opinion. Hmm.
It's like there are two currents. Well,
They have to align eventually. Let me see if my parameters are the same. Oh, look at mine.
yeah.
Yeah, no, people can't see it. Let me check my parameters. Yeah, I'm using 95 and 1, just like you. 500. Um, maybe let me see. Make the co-alignment 99.
Is that what you-
No. It makes them align like slower, but they should be more persistent.
straight up the stubborn cells?
Are you in a cell loop?
Gosh, I thought.
Yes, you are.
Oh, wait, what on earth?
Oh, what?
I don't have
Wow.
That is not, oh, if you, oh my goodness.
Oh, that was super messy. Yeah. Okay, now if it doesn't work now, I will be surprised. Okay, now put in 95 again, because we're doing nonsense.
Didn't we change?
Yeah.
Yeah, neither one of us noticed that. I guess we got distracted and then went back to it and did the same thing again.
What is this bullshit? Why are they not moving? So let's get back to it.
Where, that's the Python. I don't think it's because of this.
No. Go back to the staples. Your force persistence is 1, so 1 minus okay 0, and then why are they not moving at all? Lambda vec.
Okay, I just want to see what happens if I change this back to 20. I'm just kidding.
It's on the steppables. It's surely on the steppables, but where exactly? Force persistence, wait, self-alignment, cell velocity. Did you normalize? Yes. Huh. Angle. Let me check my code again. Wait. So, in 39, it shouldn't need, but do after cell, you have cell dot dict velocity and then open close square brackets with a column inside. Line 39. No. In the before the slash equal. Open close before the slash, open close.
You mean this?
Yes, and column.
This.
Yes.
I don't think NumPy is
Yeah, I don't think too, but you know, it's different from mine. Yeah, it's not that.
Yeah, I do that all the time with vectors.
Mm-hmm.
We need to like rubber duck therapy this silly program. Okay. Self-alignment, that was all set before.
Oh my God. No, you cannot do 44 this way because you have a minus sign on your dear X, dear Y. Yeah. That should work.
Yeah.
Okay, increase addition. See how messy it can get. I mean, some problems I don't even realize when I'm building with someone else.
Yeah. Yeah, I find it much easier to think about my code when it's just one person. I don't know.
Okay, now it's pretty obvious it's collective migration, isn't it?
Yeah.
Pretty obvious. It just takes time to form a big blob, and then all the cells moved aligned. Yeah, beautiful. It's quite impressive that such a small change can produce flocking behavior. I think this was James' idea originally. So congratulations to James. Hmm?
That's so cool.
What?
Progress in your Zoom.
Yes. And it doesn't require much to build that wound healing simulation. I will try to do that on my own. So let's hope I didn't lose too much IQ.
For every bug in your code, you lose 10 IQ.
For every month in the PhD.
Second year, so that explains why I
Mhm. Yeah, I'm feeling that already. So I will start with confluence. Confluence. With what is my cell volume? I think it's 65. Let's make this 49. And then make this a 7. Does it make sense? And then, well, we want an opening in the middle, so let's do 0 to 30. And then 60 to 100. And let's, for the purpose of seeing the stuff happening, let's reduce a lot our migration force. Okay. And okay, let's see. So the cells in closer to the cells closer to the middle, they have room to migrate. And then because they have room to migrate, they move. And then we feed back this displacement to the migration direction, and then they can close the wound. They cannot close the wound here because we lack cells. In reality, the cells would either stretch or break from the tissue or proliferate. So to see the closure happening, let's set adhesion to neutral. And we should see cells migrating to the center. Maybe.
Does skin swirl around like these cells?
Swirl around.
Yeah, like very
Ah,
my skin
got
will-
it. Epithelial cells don't move. They have to transition to another type. And then if you try to produce flocking with motile epithelial cells, they will make a tissue over again, you know. And this simulation is not working. Let me try to make them repel. Yeah, that's more quite of what we saw in the movie, isn't it? Yeah.
Still fulfilling the gap.
Yeah, but again, we cannot exactly test the hypothesis that it is the collective migration that is closing the wound. We cannot answer that yet with this simulation. But it's for a first approach on wound healing, I think that's a start. So the question from Emma is, can cells swirl around like in the simulation? If there are some bacteria movement where you increase the cell density and you see the swirls happening.
See that we grow some bacteria
Yes.
and they go.
Let's try to find it. Collective migration microscope. I'm pretty sure you. What? Where are they opening? Like new Google. I want to open the YouTube. Okay. Okay. Finally. Let's see if they swirl around. No. Oh, I can watch my own live here. No. Oh, nice. Every time you have competing migration directions, you have a swirl. Oh, this is so cute. Look at this. Does this satisfy you, Emma? Oh, that's so nice. Oh, so you can see clearly that the cell types are different. Let me close this. You see this shady region in the tip? This shady this gray area in the tip of the cluster right here. This is the lamellipodia of the leading cells. So the leading cells are clearly a different phenotype from the other cells. And they are trying to form new fronts. So these cells here, they are trying to become leaders, and they compete because they have they transmit force globally between each other.
Oh, it's like watching a political party.
I rather watch this. That's so cool, isn't it? So I don't have time, but the plan would be to show you guys the chemokinesis simulation, which I can do very quickly. Let me take the chemokinesis. And this would be Alex's job to figure out what is going on in this huge simulation here. It's not huge. It looks big because there are a lot of comments. Because when I built this simulation, I built it for educational purposes. So you can find the explanation of how it happens. And I tried to start doing annotations, but I gave up. And basically here we have a field, a linear field, constant gradient. And our cells have persistence, have a force, a migration force, and a sensitivity to the gradient. So you can see this is a linear gradient, linear field. And I'm using a trick of cell teleportation, let's say, so I don't let the cell cross the boundary. And I always see the cell going up gradient because the boundary, the field is not periodic. I mean, it is periodic, but it has this, it does like this. It is discontinuous. But I would like a infinite linear field. So to do that, I just teleport the cell back to a previous X position. That's like cheating, you know, but nobody cares. So what you can see differently from a normal chemotaxis is that this cell gets confused. You can see it getting confused. I mean, going back a little bit and then going forward like a drunk person, you know? You tell the drunk man, hey, drunk man, go grab a beer for me. And it does exactly what this cell is doing here, isn't it? It? What did they say exactly?
You said you tell the drunk man to go grab me a beer, and it does that. What is the beard?
It's, you know, I have a hard time with pronouns. Okay, overall. Yeah, they don't deserve to be treated like people. Yeah. Or it identifies as a it, you know? Yeah. Maybe drunk people identify as it. It, them. So how does this cell do that? Well, read my simulation. And then you figure it out. But differently from chemotaxis, the force in chemotaxis, the gradient in the case of chemotaxis applies a force spatially throughout the cell border. in chemokinesis, the field sensing is temporal. It's like when we try to find the source of a smell, what do? Yeah, we go around walking and we figure out if the smell is getting stronger and stronger. And then we keep going in the direction where the smell is getting stronger. This is chemokinesis. You have to have memory for that. Exactly. So this, or in other words, imagine you are trying to find the source of a sound. You have 2 ears. So you just turn your head around, and by the difference in volume or in intensity, you can figure out the direction. But if you block one ear, you have to do chemokinesis.
Phonokinesis.
Yeah. Phonokinesis, phonotaxis. Yes. Phono. Yeah, that's it. Huh?
That's the Greek one, right?
Phono? I'm not sure. Yeah. Yeah, it doesn't sound Latin. Yeah. Well, that's it for the class today. Do you guys have any question? Alex, Santhu?
No.
No.
No? Okay, so let's get out of here. The class ended. I see you in the next week. Finally.
Yes.
What?